Grazie mille, buongiorno a tutti. Maria Luisa, molte grazie. È un grande piacere per me partecipare in questo congresso. While I speak Italian, my Italian is, because of my ancestry, my Italian is a little rusty, so I will not do as Serge did. I will proceed with my presentation in English. Just for disclosures, after 10 years at Amgen, 1 year ago I moved to UCB, where I am now leading the project of sclerostin inhibition in its clinical phase, a project that you may know is being developed as a collaboration between Amgen and UCB. At the center of the story of sclerostin inhibition is an experiment of human nature. And through the characterization of a few hundred individuals in the world that were identified because of their phenotype, that as seen in this slide is characterized by increased bone formation, thick bones, and an historical resistance to fracture, the discovery of sclerostin came about. And because of the fact that in these patients, depending on their being homozygous or heterozygous, their levels of sclerostin production is different from being null in the homozygous to having different levels of sclerostin reduction. The hypothesis moved forward that the effect of sclerostin in bone biology could be modulated to indeed have a controlled impact on the skeleton. As you probably know, sclerostin is a protein primarily synthesized by osteocytes, and sclerostin functions as an inhibitor of bone formation. And it does that by binding to the LRP4 receptors in the osteoblast surface and in pre-osteoblastic cells. And by doing so, it prevents the binding of frizzle and LRP4 and 5, and therefore shutting down the bone formation abilities of these cells. When one captures or inhibits sclerostin, such as can be done by a sclerostin antibody, shown in this cartoon, then frizzle can go on to binding with LRP4 and modulate the chain of events intercellular and internuclear that lead to increased bone formation. So as it is in clinical development, in order to prove that the concept works, we usually work with animals, and in these experiments conducted by Amgen and UCB and published by Lee, we see that when you knock out sclerostin, indeed the phenotype is reproduced, and as you can see in male mice or female mice, in trabecular bone or in cortical bone, the increase in bone mass, density, and microstructure is significant by the absence of sclerostin. This increase in bone density, mass, and microstructure is largely the result of a very significant increase in bone formation, and as you can see in the bottom panel, in both trabecular surfaces and in cortical bone, both at the periosteal surface or at the endocortical surface, there is significant increase in bone formation, as can be seen in this double label staining. And this corresponds to significant increases in bone formation surface, in trabecular bone, and in cortical bone, as one would expect from just the visual impact of the experiment. More importantly, these improvements in mass, structure, and density are accompanied by improvements in compressive strength in the vertebral body, or the typical 4-point bending test, meaning that the improvements in strength, which mine are above that of erectomized animals or sham animals, are significant and represent bone of significant quality. After a series of preclinical studies, clinical studies are moved forward, and this is the schema of our phase 2 trial, which was already published in the New England Journal of Medicine last year, and resulted from testing several doses of romosozumab, a humanized monoclonal antibody against sclerostin, provided subcutaneously once a month or once every 3 months. In this particular study, in addition to having a placebo arm, we also had an open-label alendronate and an open-label teriparatide arm. And for simplicity, I will only present in this 10 minutes the results of the romosozumab 10 milligrams, 210 milligrams given once a month, and the corresponding control groups. And the reason for that is that 210 milligrams once a month is the dose that we have chosen to advance in the phase 3 clinical trials. You may be familiar with this data. This shows in the top line the increases in density of the lumbar spine over 12 months of romosozumab administration, achieving approximately 11.5% by 12 months, which correspond to actually slightly over 1 full T-score. And these increases in density were occurring rapidly and were more robust than those of the comparators, in this case teriparatide, alendronate in the blue line, or of course the placebo calcium and vitamin D treated subjects. Similar results were observed at the total hip and the femoral neck, as observed in these panels. And notice here that again, the changes in density, the improvements in density were significant at 12 months and 6 months compared to all comparators. And note also at the hip, the similarities between the changes seen with alendronate and teriparatide, something that perhaps should not be too surprising. These significant gains in bone mass density are the result of a peculiar and interesting profile in bone turnover. In the left panel, you see P1NP, a marker of bone formation. In the right panel, you see CTX, a marker of bone resorption. And in green, the effects of romosozumab to increase rapidly and significantly bone formation, with a pattern that returns towards baseline by approximately 6 months when measured by P1NP, and a pattern simultaneous of decrease in bone resorption measured by CTX, which remains reduced throughout 12 months of therapy. For comparison, the typical increase in both formation but also in resorption known to occur with teriparatide, and the typical response of decreased bone turnover, formation, and resorption with the anti-resorbers such as alendronate. From a standpoint of adverse events, we cannot say too much because this was just a small study, and next year the results of the first large over 6,000 ladies on romosozumab versus placebo will be available, so we'll know more about the safety profile. But to date, the drug has been well tolerated with an imbalance in the adverse events at the site of the injection, which usually are temporary and are characterized by mild induration or mild pain or itching or an injection site hematoma. Just to be balanced with other available data, last year the Eli Lilly group presented their data on development of a similar antibody. There is blosozumab, and they also showed a dose response, and over 12 months show similar improvements in density, depending on which dose you look at, than those that we observed in our trial. Eli Lilly at present has put on hold the development of their antibody. As it is done with most phase 2 studies, the phase 2 study in the first year that I just showed was followed by another year of administration of romosozumab. I'll show you the results in a second, after which we evaluated the conversion, the basically follow-on of a anti-resorbic therapy, and for this particular study, we chose to follow up with denosumab or put the patients on calcium vitamin D to determine the effect of discontinuation of romosozumab. And these are the effect of the discontinuation. After 2 years of therapy, mind you that in the second year there are continuous improvements in density at the spine and at the total hip. Nevertheless, these improvements are not of the magnitude that were seen in the first year. After you discontinue romosozumab, as it is with most therapies for osteoporosis, there is a reversal of the gains in bone density that had been observed. Those can be maintained or perhaps improved upon by a follow-on therapy with an anti-resorber such as denosumab, as seen in this slide, both at the spine and at the hip. In conclusion, in animals, sclerostin inhibition results in increased bone formation, decreased bone resorption, and increased bone mass and strength. In humans, the administration of sclerostin antibodies leads to substantial and continued BMD increases up to 2 years of therapy through an increase in bone formation and also a reduction simultaneous in bone resorption. These effects are reversible upon discontinuation, but may be further augmented or maintained by follow-on anti-resorptive therapy, and overall, to date, romosozumab has been well tolerated. Just to confirm that this program continues and has its phase 3 program ongoing, and the results of the first of the large studies will materialize in the first part of 2016, and we are looking forward to sharing those results when they become available. I again thank you so much for the opportunity, Maria Luisa, and if we have time, we'd love to entertain some questions.